And now, fundamentals of data analysis, which is the second lecture learning in our class this semester. So I'm going to focus on 7 principles. These are principles that are the absolute minimum concepts you need to start doing data analysis. I want to point out that some of these things we'll cover again and again. So if you see repetition in the class, you're right about that. I want you to get these things. And so we'll return to these principles throughout the semester. Here's the most important first principle. I'm not sure if I've said this in class yet, but I will say it many times. As a scientist, investigator, data handler, etc., it is your personal responsibility to make sure that the data analysis you're doing is appropriate. If you have a collaborator, a statistician, a PI, a mentor, that's great. It is still your project. If it's your honors thesis, if it's your master's thesis, if it's your PhD chapter, it's your responsibility. You're going to have to take that responsibility and ownership. That's the first principle. It's also not something you can throw together at the last minute. It requires a long-term commitment. This is a fundamental issue. Students who don't know about data analysis yet oftentimes think, in the same way they think about other classes like psychology or history or something, that you can wait to the last minute and do it. The night before the test, I'm going to study. It doesn't work that way in data analysis. You're almost always going to have a poor outcome if you do it that way. It's something you have to work on and think about and work on and think about. And if you get involved in a master's thesis or PhD kind of project, or if you're a technician or something, you're working on a big project, you need to start thinking about your data analysis on the scale of 6 months, 8 months. That's how long it's going to take. And if you get into this mode of like, I've had students before who are like, they started their data analysis. It didn't work very well at first. They start to cry and they're like, I can't do it anymore. I might start to cry, by the way. I'm not saying crying is bad. And I'm like, oh, that's interesting. Like, when did you start? And they're like, oh, Tuesday. And the meeting is on Wednesday. I'm like, you're very confused. Okay. You start on Tuesday of September. Before you need to work until July. And then we can start talking about it's not working. That's the scale. And so you need to understand this early on. I've oftentimes told my honors students that you should be deep into data analysis by November of your senior year. November. That's for an honors thesis. If you're a master's student, you kind of need to start data analysis your first Christmas. I mean, absolutely as soon as possible, start doing data analysis. So it takes a long-term commitment. One of the theories for the class, and the reason why I will give you a 0 if you turn your stuff in late, is because you must not adopt the habit of waiting to the last minute. We cannot have this thing where like, well, I thought it was due at 11. I got something that I assigned to you weeks ago, a month ago. And then it's like the last night. And you're like, well, I forgot. It's not going to work. You need to be working on that 2 weeks ago. You got to work ahead of things. So I'll stop there. This course and any other course you can take will only give you doorways or concepts in data analysis that you can apply. It's going to ultimately be up to you to verify, explore, execute, and present your results honestly. It's no one else's responsibility. It's your responsibility. This is why I told you before, if you want to be a scientist, you need to get this book to get started. Like if I want a career in science, this is not enough. You need probably multiple books. And you're going to need to be willing to be like, okay, I'm working with proportions data. So I'm going to get this book out. I'm going to go like, okay, I'm going to spend the next 3 or 4 days reading about how do you analyze proportions. And then I'm going to start running some code. Okay, this is how you analyze proportions. That's interesting. use some examples from the internet. A month later, maybe you kind of understand the right kind of code to analyze proportions. But you can't sit there and be like, oh, I got to wait for my PI to get back to me, or I got to email. Who knows how to do this? You do. You have to figure it out. Do you have a mirror? You can use your phone as a mirror. I figured this out. You can like flip it, get that, and look in that. This is the person responsible. It is up to you to do this work. And it is ultimately a test of your personal ethics, whether you're willing to own your data analysis. This is something that my PI a long time ago, my postdoc mentor told me. And I was like, oh, that was a big moment for me. It's just like, you have to own it. Whatever it is, you have to own it. You can't present a figure on a slide at your defense, that's your data, and be like, I don't know what happened. You have to own it. If there's errors, you own those errors. You just say, there was an error. Susan was supposed to pick up the seedlings and she forgot. And then 2 weeks passed. And so that particular data set's 2 weeks late. That's okay. You're being honest. Et cetera, et cetera, et cetera. Okay. I'll come back to this over and over again. It is a broken record for the class. Second principle. You need to understand the biology of the system you're working in and the inherent sources of variation. This idea of variance and variation is something we will return to again and again throughout the semester. So, one idea is frequency distribution. What is the frequency distribution of the trait in your study subject or in the study system? Do you have a distribution that looks like this? See my cursor? This is a normal distribution, so-called. This is the first time I've said that in class, maybe. You've got a mean value, and you've got tails coming away from that mean value that are more or less symmetrical. This would be like weight. That's what this example is actually. This is 400 pounds. This is 100 pounds. I don't know if this is males or what, but if you like measured all the human beings on the University of Dayton campus, all the whatever, you would end up with this kind of a distribution, a symmetrical distribution. And you can predictably do analysis with that underlying assumption, a bell-shaped assumption. If you're dealing with human beings' weight, you know that this is a distribution. But some other distributions have a different shape. For example, income. Income, you might have this kind of shape. This is like the mean or median. I don't remember where I got the data. Annual income is 25,000, and then it tails off. So that's a skewed distribution. That would be called skewed right. What is the underlying distribution or the frequency distribution in your study subject? You could also have a bimodal distribution. The biology of your system will dictate this to some extent. If you don't know, if you're a little bit confused about what I'm talking about, this is another example. This is from my class. I used to teach ecology. My class, that particular exam, there was a mean of 78, and it had this distribution that was skewed right, and had this little blip out here. So a few A's, a whole bunch of B's, by far the most highest, 80 to 90, whatever, in the B range, and then skewed right. This is just another example if you're having trouble thinking about a frequency distribution. Now, let me just take a moment here to give you an example. You can have a distribution like weight, or like the length of your finger, or the width or the depth of your nail depth. I could measure nail depth across campus or something. And again, you would come up with a reliably bell-shaped distribution, because some biological traits have a reliably bell-shaped distribution. You need to understand this. If I was dealing with not your nail depth, but if I took a swab from under your fingernail and did an analysis of the microbial community, we could have radically different variables. It would not be bell-shaped, probably. You would have some extreme values. I was just scratching my dog just right now, a minute ago. So the bacterial community or whatever under my fingernail would be through the roof. It would be millions. Whereas somebody who washes their hands a lot or uses a gel, you may have nothing up there. So this is an example. So nail depth is a reliably bell-shaped distribution. Microbial community under the nail is in no way bell-shaped. You have every reason to believe it will be radically different. So you need to be thinking about this. Normal statistics might not work for nail depth. They definitely would, but they would work for nail depth. They might not work for the microbial community. Let's see. Can you see it? There we go. See those microbes? Here we go. You can probably look at it this way. See those? That is, whoa. Another example, if you're measuring export from a watershed of nitrogen, what that means is how much nitrogen is coming off into a stream. Like, okay, I'm an aquatic biologist. I want to measure how much nitrogen is coming off into stream nitrogen. It's an important topic. In a lot of ecosystems, nitrogen export or phosphorus export, the things that drive algal blooms, which is a big environmental problem, are very tightly linked to pulses that are linked to weather events and anthropogenic disturbance. So specifically, if you go out and you measure nitrogen in a stream, just right now, today, maybe like, oh, there's not very much nitrogen in the stream. You could measure it in 100 days of the year and be like, not very much nitrogen in the stream. However, if you measure it after a rainstorm, after the agricultural farmer folks have put their fertilizer down, you might have, 10,000 times the amount of nitrogen in the stream because they put the fertilizer down, it rained, it flushed off into the stream almost, especially for phosphorus, which is a geonutrient. Almost all the phosphorus in the whole budget comes off of ag fields. And that happens because of they put the fertilizer down, there was water, rain, so runoff. So if you measure the stream, just like today, January, you'd be like totally misunderstanding what's happening. You need to understand the biology, back to the main point, you need to understand the biology of the system you are working with and the inherent sources of variation. In a stream system, an inherent source of variation is temporal or time in relationship to anthropogenic activity and weather events. For fingernails, it has to do with whether you scratched your dog or whatever, for example. So you need to understand that to set up your study correctly. Third principle, your experimental design, and specifically the relationship of your sample size to the variance will determine your ability to answer the questions in your study. Again, all of these topics, each of these things is very deep, and I'll come back to them again and again. This is just introducing you to these ideas. I encourage you to think about each of these in some depth. And I'll return to this equation later when you start doing analysis, but I just want to point it out here. Sample size, so you're going to be calculating variance. Variance is a key issue. How much variation is in your system? And we're not going to do this like with a pencil and a notepad, but this kind of calculation or something similar underlies all statistical tests, something like this. And in this variance calculation, sample size is in the denominator. It's here, see this? See my cursor? What this means is sample size is mathematically related to variance in such a way that it reduces variance as it goes up. As n goes up, variance goes down mathematically. Sample size is key to understanding your system, especially in relationship to the amount of variance in the system. You may have something like nail depth, where we've got a pretty darn reliable frequency distribution. We know, I could go measure it, get out of it. Or you could have something like microbial community under there, which is wild variance. You need way bigger sample size to capture that variance. So I'm going to meditate on this with you for a minute on sample size. In data analysis, there is signal and there is noise. Signal is what you're trying to accomplish, or what you want to get out of your study. So like, I want to measure tree seedling growth in relationship to microbial soil microbes. Just as an example, soil fungi. And so the signal in the study that you're trying to hear basically is, does the tree seedling respond to the soil fungi? I'm going to put some tree seedlings, I'm going to dip them in soil fungi, put them in the field. That's signal. Noise is all the other stuff. Beetles eating the tree seedlings. Rain happens. A deer chomped it. Somebody stepped on it. It was not planted deep enough. There's all this noise. So every study has signal and has noise. You got fish in a tank, okay? Oh, I got to see if these fish, how they're responding to this scary thing that's happening. Okay, well, the lights on or off? What's the temperature in the room? Was there noise in the room? Did somebody accidentally drop a little Gatorade into the tank when they were taking a drink? All this stuff can be noise. Also, there's a sort of external factors. There's also internal factors that are noise. For example, genetic variance among the tree seedlings, genetic variation among the fish in the tank. This is noise in the system as well. Mathematically, sample size in this equation, don't get too worried. We'll come back to it over and over again. N is the number of samples in the denominator of variance estimates. And so technically, more samples helps you reduce the noise because it reduces the variance. The classic blunder of a beginning scientist or an experienced scientist who is entering the world of experiments and data analysis for the first time is the creation of designs that have too few replicates. This is the problem that a lot of young scientists or inexperienced scientists who are, just trying an experiment for the first time, where they make a mistake. You have to ponder deeply the sources of variation in your study system, err on the side of having too many samples or plots or replicates, not too few. Simpler designs with greater sample size of fewer treatments are generally better, especially when you first get started. What happens is, especially young scientists are frisky and they start thinking about all these different things. Let's have this experiment that has all of these factors. But the more factors you have, the bigger the study becomes. The bigger the study becomes, the less space you have. I don't have that many tanks. Don't have that much space in my greenhouse. I don't have that many, salamanders that have been genetically modified, or frozen frogs, or whatever. And you go, well, I can add a treatment, but I have to reduce the sample size to make it fit. This is the blunder. When it's time for your statistical analysis and you want to compare groups, what you are ultimately doing is comparing differences among samples in relation to the variation within the samples. Especially if you have high variation within your samples, the microbes under the nail. You need more samples. More samples reduces variation and makes the signal clearer. This results in an increased likelihood that you can ask the question that you set out to ask. And this is what I want for you. That I want you, when you finish your study, you've left here, okay? You're at University of Kansas, and you're doing your PhD, and you're on your first chapter. I want you to be able to publish your data. have real data, not for it to be a year wasted, thrown in the trash can. You, not me. It's got nothing to do with me. I'm in UD. I don't even know where you went to. Okay. This is about your empowerment. I want you to be able to answer the question you set out to ask. That's the point. When you have very small sample size, statistical tools will not work. And you will be SOL by design. And that's what we're trying to avoid. If you make your sample size large, if you get the design correct, the rest is, you're okay. If some reviewer or your PI or a committee member goes, hey, you didn't do that right. You should do a mixed model ANOVA versus, or whatever. You should do multiple regret. Whatever they say, you can be like, it's fine. I don't care. I can do that. I can solve that. What you can't solve is you don't have replicates in your study. You don't have enough replicates. If you make your sample size reasonably large relative to the variance in your system, at least you can pound in that square peg. You can make something good happen. One last thing. There's a tremendous difference between a study that yields results which fail to reject a null hypothesis and a study that yields results that is too vague to come to a conclusion. If you can confidently say drug A and drug B have the same effect on infections, you fail to reject the null. So, okay, I'm going to give this person this COVID drug, not this person, this subset of my study. I'm going to apply drug A to this 1,000 people and a drug B to this other 1,000 people. Compare the two, no difference. That's good science. You can say there's no difference between these two drugs. If your results are, we don't know if drug A and drug B are different because our statistics are too messy because we screwed this whole thing up. That's being SOL by design, and you want to avoid that. Don't be SOL by design. Fourth principle, honest and clear data display is a foundation to good science. So we're going to take a little journey in this now. I'll give you some examples. We'll do this in the coding club as well multiple times. Here's an example of a bar chart. We'll talk about this at some length. You should really only use a bar chart when you don't have variance in your system. You're just trying to display a number. Like, how many wins did the University of Dayton have in the last 5 seasons? 22, 24, 26, 18. All right, there's no variance there. That's a number of seasons. And so if you're like, I just don't want to put that in a table, I want to display it, you can do a bar chart. That's fine. If you've got variance in your system, then you shouldn't probably ever use a bar chart. We'll come back to that. But here's a bar chart with some data. This is real. This is from a student a while back. 1989, 1999, 2012. And you go, okay, look at this. There's a big difference between 1989 and 1999. This difference seems like a lot. Here is error bars, because this actually was a study where we could calculate error. There was replication. You go, okay, here are error bars, and I'll show you or talk about how to calculate those at some point later. This is probably one standard error or one standard deviation around, and these values are the mean, most likely. And this is what you're going to have to figure out when you're starting to do data display, and we'll spend the whole semester working on this. What is this? What is this? What do these two things mean? Okay, in this particular case, this was the mean and this was the error. Now, error, the way you understand and think about an error bar is, do the error bars overlap? And it's symmetrical, so it goes down here. I can't even see it here, but the top of this one and the bottom of that one are kind of on top of each other. These two are definitely on top of each other, so it becomes less impressive if you add error bars to it. Okay. Now, one thing I want you to think about anytime you look at a figure from now on until for the rest of your life is, what is this? Do we know what the x-axis is in this data display? Well, it's year, but it doesn't say year there. That would be helpful. What is this? I just told you it was mean, but anytime you look at a figure, you should ask yourself these questions. What is the x-axis? What does the top of the bar mean? Is it mean, median? Is it a count? What is it? Your presenter should tell you that. And if you're presenting, you should tell people that so they know what that is. What is this? It's an error bar, I just told you, but I should tell you that it's one standard deviation or one standard error, what that means. Okay. Now, I've taken the same data set and I've rescaled the y-axis. See this y-axis? It's going from 20 to 40. Okay. If we go to 0 to 50, now this starts to look less impressive. It's still a slight step down, but it starts to look like the same thing. This is a bit more honest data display. And here's a box plot. And now I've got sampling year. I've got coarse woody debris biomass. So now we've explained both of the axes. And instead of using a bar, I used a box plot. And we're going to work with box plots the whole semester. And I'll talk about this at length. Let me just give you a quick one right now. This center bar is the median. This is what's called the IQR, interquartile range. 75% of the data are captured in this box. The whiskers is the extent of the data set, except for outliers. Outliers are these points here. This has a lot of outliers. There'll be a long conversation about outliers coming up. But an outlier is something in the data set which is mathematically far outside the range, depending on your box plot calculation. And I'll explain that to you later, the actual math of it. But usually it's like 3 times the IQR. So you're way outside the range. And so this is weird. They're weird points. Actually, all of this whole data set has a whole bunch of outliers in it. So this looks really messy. I'm going to talk about data interpretation this whole semester. Instead of so what you've done here is by going from this, whoa, this is a lot. This is kind of dishonest data display. This, which is very honest data display, not quite what we're going to get to in this class, but it's good. So nothing here. I mean, this box is slightly lower than the other two, but look, there's a massive overlap in the data sets. Sorry, there's nothing here from a statistical perspective. I realize this is in the weeds, but that's what you have to do with data display. You've got to go in the weeds. You have to think about every single one of these things. What's your y-axis? What's your x-axis? What are the labels? What are you putting in here? What is all this stuff? You have to think about all that stuff carefully. I want you to commit to honest data display for the rest of your life, starting now. Okay. The next thing. A fundamental commitment to exploratory data analysis and other background activity is required for sound statistics. Now, this is not clear to you yet because you've never heard of these things. What I mean here is ultimately, you're going to want to run a formal test. A formal test is going to generate a p-value. And the p-value is what you're going to use to say whether your results are significant or not. Have a statistically significant result. Your PI and you, when you're doing your thesis, you're going to want to get to that. Like, I got to get to this, come to that point. I am telling you, this is, again, a core principle of the class, something I want you to adopt for the rest of your life, is you have to start with fundamental commitment to exploratory data analysis, which leads us to the concept of doves. Before you do any analysis of your data set and start chasing p-values and statistical results, t-tests or NOVA or whatever it might be, you need to look out for doves. This is something I came up with. You won't find this anywhere else. This is just a RightMQ, it's special. But I think this, if you do this, you will become empowered. And I don't mean just for your own study. I mean, anytime someone tells you that they've done an analysis and arrived at some result, you can use this way of thinking to determine whether you think they're right or not, whether you believe them. Doves. What are doves? Distribution, outliers, variance, effects, and sample size. Every data set you see for the rest of your life, start by looking for doves. Distribution, outlier, variance, effects, sample size. So you're going to scan, before you do any formal analysis, what you're going to do is scan for your data and think about this from the perspective of doves. What's happening with your data set? And you can scan through your data using many different techniques. You're not looking for p-values here yet. You're just trying to understand what's happening in your data set. So frequency distribution, like this, a plot, box plot kind of comparison like this, which we'll be doing this. All this stuff we'll be doing in class. A time series, something like this. This is actually from a study I published a while back. Look at these little error bars right there. Regression kind of approaches like this. I'm telling you, before you go like, I need to run an ANOVA on my data, you have to scan for doves using visual displays and using statistical displays or statistical analyses. One great way to do this is to, if you're comparing groups, is to create a strip chart, or I'll call it a dot plot in ggplot, because that displays every single point in your data set. If you use a dot plot or and a box plot together side by side like this, you can see both the effect size, also the distribution of the data set, whether the sample sizes are equal among them, what's the range. The box plot will automatically identify outliers. See these box plots always, no matter how you calculate them, generate an outlier analysis, and it tells you that these top 2 points here are considered mathematically outliers in this data set. You have a lot of information here. You can overlay these, and I'll show you how to do this to make a fourth plot, which looks like this. Now I can see the box plot. I can see where the medians are. I can see where this box relates to this box. I can see what the sample size is. I can see if there's any outliers. Again, there's 2 outliers in this data set, but they're right kind of at the tail of the distribution, so this doesn't worry me much at all. You can see whether there's a bimodal distribution or whether it's more or less of a normal distribution. You can see a lot of information. Here's a kind of figure that we published recently. My doctoral student, Michaela, published this. So it's a peer-reviewed journal article graph, and it's what I just described. So this is a box plot, and it's got this overlaying dot plot or strip chart, which is together. I call this a fourth plot. And you can see there's one weird thing up here, but more or less, you can see what the distribution is. You can see these medians over here. These 2 medians are quite a lot above these 2 medians. Right here, you can see what the range or the distribution of this data sets is. You can see what the sample size is. You can see distribution. You can test for outliers. You can think about variance here. You can see the effects. The effects is these boxes are higher than these 2 boxes. Sample size, you can literally count 1, 2, 3, 4, 5, 6, 7. I know how many samples are there. Everything you need is there on the screen. And this is honest data display. If you've planned your experiment correctly and properly considered sources of variation, and then you search for doves, that will reveal key information about your study system. If you have not planned properly and you have issues, sample size and variance, this is what it sounds like when doves cry. That's not what you want. If I was a good teacher, this song would start to play right now. I'm not. So you just have to imagine it. You don't want doves to be crying in your study set or in your study data. Okay, and we're going to hammer this doves thing the whole semester. until you just do it automatically. When you're like at a seminar, you see somebody present, you see your fellow grad students present, you're just like, oh, doesn't look like, that does look like it's crying there. Sixth principle. Your analysis has assumptions and limitations. You need to know what those are and test them and communicate them. Every statistical test has assumptions. It's not fun to do the work of testing the assumptions. You're going to want to speed through this. You're like, oh, I scared these fish. I measured their cortisol. I want to know whether it's higher or lower, so I'm going to run a t-test or a NOVA or something. Just run it. But, oh, look at this great result. Okay. If it's exploratory data analysis, go for it. Rock it out. There's no rules. Do everything you want. If you're presenting that at your thesis defense or you're putting it in your paper, you better have tested the assumptions, because a NOVA has a whole bunch of assumptions. You can't just run it. In R or any other statistical package won't stop you. It will generate a p-value. You just hit go and it goes, here's the p-value. Sometimes there might be a little red flag there, but a lot of times it's just like, here you go. And you're just like, okay, great, got it. Except the problem is you didn't test your assumptions, and that's an invalid test. And now you've got hogwash, not science. So you're going to have, and this is the reason why I told you need to buy this book, even to just get started. Because books like this will tell you what the assumptions are. And it's not you might not think it's the most pleasant thing to do, like on a Sunday afternoon, to be like, I'm just going to flip through here. Let me just review the assumptions of multiple regression. It's required, though. You have to do it. And you've got to get in the habit of being like, if I'm going to generate a p-value, I need to test the assumptions. And we're going to do that in our class. But it's something you have to commit to. It's a core principle. A lot of times, statistical analyses will assume a normal distribution. So you have to test. Does my data meet the assumption of a normal distribution? We're going to do this in class. And the seventh principle is that statistical significance is not the same thing as biological significance. And it is on you to communicate the meaning of your analysis within context. So again, I'm asking you to do exploratory data analysis. I'm asking you to present your data honestly. I'm asking you to search for doves. At some point, you can communicate the results of your study just that way. Your PI and you and the journal will also want you to generate a statistical test, a p-value. And we're going to do that in class for some data sets. And those are legitimate. I'm not against those. Those are good. You need to do that. But you may have a statistical result that you're like, where it seems to be not significant. Does that mean there's biological significance or not? So here's an example. If you had some illness, okay, that was like a terminal illness or something, this is going to go really depressing. There's some kind of cancer or something. It's just like, oh, I'm going to start to crap out here. I'm going to die. And you go, okay. Some scientists did a study and they go, okay, this drug A and drug B, I did the test statistically, and I did some experiment. Statistically, they're not different. However, drug A, on average, the person lives 6 months longer. Are you just going to be like, well, guess nothing happened? No. The biological significance to you might be, let's go with the 6 months one, whether or not the p-value is whatever. This is a kind of a deep topic, and we're gonna spend a lot of time in it. But I just wanted to put that as a main core principle of the class. Biological significance is not the same thing necessarily as statistical significance. Okay. So I'm going to spend a little bit more time on this topic, exploratory data analysis. Okay. Actually, I'm going to stop here because I think this is probably too much. Just 40 minutes is probably too much Zoom. And I'm going to break this out into a separate its own separate video. Okay, actually, no, I'm not. I'm going to keep going because you can just pause. So let's just roll with it. Okay, a principal task for early data analyst is committing to exploratory data analysis. I mentioned that as one of our core principles. That was principle number 5. You can't skip 2 year discrete statistical tests. You have to do this. You have to do exploratory data analysis. In some cases, you may have an idea from the very start of your study what you're trying to measure. Like it could be very, very clean. It's like, I'm just going to measure this and I'm going to run this test. But in most studies, there's other things happening. There may be many variables that have been measured. I measured soil, I measured, temperature, I measured whatever. Lots of different things about the fish tank or about the insects or whatever. Sometimes your study takes an unsuspecting turn, focus will shift, maybe something happened, you discovered something early on, you got, this also run this test, et cetera, et cetera. Even if you have a pretty good idea of what you're getting at, what comparisons you want to make, exploration of your data is highly advised. It's the 5th principle of our 7. So to do exploratory data analysis, I want to talk about how to make a nice graph. And this is from Whitlock and Schluter, chapter 2, how to draw a good graph, show the pattern, show the data, make patterns easy to understand, represent your magnitudes honestly, and make your graphical elements clear. We talked about some of this already, but I'm just going to journey through some different kinds of charts and graphs. So this is an example of a bar chart. In this particular case, I did publish this. Height of this magnet, this is actually species per acre, which is a weird variable. There is not variance in this. This is just 5 different sites. We don't need to estimate variance around those. Here is a bar chart that has error bars and statistical significance. This is higher than this. This is higher than this. Okay. This is a bar chart with error bars. This is a good way to get feel for a variation around a mean. So this value would be the mean, and this would be potentially 1 standard error around that. A rule of thumb is if the error bars overlap, the bars are not significant. Borth plot, I already mentioned this is a good way to display data. We will focus on this. This is good for exploratory data analysis. This is a good standard practice for comparing groups. This is a way you can see doves in your analysis immediately, and you can present those to your reader or to your PI or to whoever's in the audience. When you have lots of data for exploratory data analysis, one thing you might want to do is make scattergrams. Scatter diagrams. We'll do this very soon in class. So you can compare the magnitude of one variable versus another in a linear fashion. As one increases, does the other also increase? And you can think about the strength of the line. Does this seem clear to you? How much scatter is in the data set? What's your sample size can be displayed this way. When you're doing exploratory data analysis, making a bunch of these things is a good idea. Sometimes your variable might be a curve. So we'll do this in class as well. These are curvilinear relationships. See this? This one looks a little bit floppy to me. I don't really like this. This one looks really clean. So you can display this and you can think about it. So you might have variable, you're like, there's a relationship between these 2 variables, but it doesn't seem to be a straight line. It seems to be, there could be an inflection point. There could be thresholds in your data set. You need to understand what these are. If you're going to try to run like, I'm going to run this big statistical test, but you've got some variable in here that's got this curvilinear relationship, it could mess the whole thing up. So you need to know beforehand. But to find out beforehand, you need to do exploratory data analysis and figure this stuff out. You can do big panels like this. I'll show you how to like owl stuff up like this in R. You can block them together to create a figure for your manuscript, but you also might just want to be doing this as a way for you, before you present it to anyone, no one may ever see this, but it's just like, I need to get my head around the relationship between copper and aluminum, magnesium, and whatever, organic matter, manganese in my data set. Like, I need to understand all these relationships before I can formulate a really strong statistical test. You can make big diagrams like this and drive lines through them. This is not that hard to generate in R. I'll show you at some point, maybe towards the end of the semester, how you can make these, like, just throw all of your relationships up at once, and you can just like visually scan through them. Like, if I just wanted to, again, not as this is a statistical test, we're not presenting this necessarily in our manuscript, although you might, but just before you go meet with your advisor, you go like, okay, I'm going to throw all this up here and just like snapshot what's going on. And if, just for me, quickly, I like, this seems like something here. See this? Oh, I lost my pointer. Here we go. This seems like something here. This seems like something here. Some of these lines seem to be kind of flat. This looks like there might be something here. I can see the R squareds right here, 3, 2. Negative 5, 3, this is a strong negative relationship. So you can start to think about your study. And there may be multivariate relationships among these things too. But you want to look at univariate, univariate, univariate. So you like, I understand everything that's happening. And let me just pause here if I haven't said this already. One thing you need to do if you want to be truly and fully empowered is get into the habit of generating. I would say literally print this stuff out. Like, when you start your analysis, print this out and take the sheets into your advisor's office. Like, oh, I've got 4 graphs here or 5 graphs, and like put them on that person's desk and like push them like, hey, let's look at this together. And it's a scary thing to do if it's your study, because you're like, oh my God, did I do it wrong? But this is a way for you to become empowered, because your advisor might immediately go, oh, here's something I see that we need to explore further. Do a second analysis like this, like 0.53 FDIS and AGB residuals. It's a complicated analysis. Don't worry about that part. But like, this 5.3 is something we need to look at more carefully. Like this, try to figure this out. It's like zoom in on this one and spend time on that. And you should start doing that early. This is what before I said, like, you need 8 months, 6 months. It's true, because the most exciting thing in your whole science might be buried in this, and you don't know if you don't explore it. Here's some more scatter diagrams. So in this particular case, I didn't drive a line through these. I didn't run statistical tests. I just presented this. This is published in a review journal article, because what I wanted to show my reader was, see all these points right there, and then this is way higher right here. See all these, this thing is like up, down, and up, down, and up, because the theory for these trees, these are oak trees, underlying theory for these trees is that they're shade intolerant. They have to grow in full sun all the time, but that's not true. This thing grew from almost 1650, almost 1750. That's 100 years suppressed, and then grew fast after. Just this, like Occam's razor for like a Rorschach of this, tells us a lot about what's happening with these trees. So this is elegance and simplicity in data display. Don't be afraid. This is a different kind of scatter diagram, the date and sample diameter. And now I've colored the points here, trying to display something about the about my data set. This is like just letting the reader see my data set. So you can see the total sample size, all the things you get from this figure. The total sample size, you count these 1, 2, 3, 4, 5, 6, 7. Okay. You can go, okay, which of these, what's the size of my samples? Okay, some are this big, some are this big. When did these, and these are tree cross sections, when did these trees start growing? How old are they? Okay, some of my trees were 1860, the most recent ones are 1960. Okay, what's the relationship between sample diameter and date? It's a negative relationship, but it's I mean, there's a strong negative relationship, but there's a lot of variance around that line. If you can just imagine drawing a line through it. Okay, also, gray points are historical, there are kinds of fire scars, and we're going to actually work with this data set in the class, not make this figure, but work with the data set. Okay, how many of them have historical prescribed fire scars? That's the little pluses, or both. I can see that here. So this is just a way of opening up my data set for a reader or somebody in an audience to understand almost everything about what's here. This is honest data display. Now you see everything I've got. Again, it's scary, especially if you're like, ugh, my study is messed up, or you might be afraid. But this is what you have to commit to. Remember, I told you my postdoc was like, you have to own this. You have to own your data. Owning your own data analysis is the fundamental question of your personal ethics. So this is being as honest as possible. Now you see everything about my data, or at least about the samples. The data analysis comes second. This is everything about the samples. This is a time series figure. So this is day, and this is germination percent. These little things are error bars. So if you see error bars, there must be replication. This point must be the mean or median or some measure of central tendency. In most cases, if you've got error bars, see my cursor? That point on this axis is the mean for some treatment, and around that is the variance, and usually that would be one standard error. We'll calculate that later. So you're like, okay, this is the mean and this is a variance, and this is pretty tight. I mean, these error bars can get really big. Here you see error bars overlapping. This error bar is way separate from this one. So this looks really different from this, and these two look really different from that. Okay. So you might want to create a time series like this. This is a time series. It's very common in biology. It might be, oh, I'll put these fishes in a tank, and 5 days later, this is what happened, or whatever. Oh yeah, just zoom in on the error bars. Stacked bar chart. I don't know if we'll do this in class, but maybe. This is a good way to display data. A lot of times with molecular data these days, people are using stacked bar charts. Here is, in this particular case, this is the species. So these are tree species. This is different oak trees: white oak, black oak, red maple, sugar maple, chestnut oak. And the percentage of the bar that's colored in this color is how much percentage that species makes up of the relative importance in the overstory of this forest. Okay, well, that may be too complicated if you don't do forest ecology, but you can imagine that you sampled the microbial community on different surfaces or something. And now you've done some big molecular analysis genetic analysis to know what, it won't be species in that case, it would be like functional groups or something, like the Firmicutes or something. And you might want to say, okay, on toilet seats, this is the most important, most dominant one. And on doorknobs, this is a totally different set of bacteria, whatever. You might want to create stacked bar charts for your exploratory data analysis. You could do bar charts that represent change. This is like effect size. You can have, this is negative, this is positive, and there's these asterisks. The asterisks represent statistical significance. A lot of times on a chart, you'll have some kind of bar and then some estimate of statistical significance. This is something I published a while back. You can mix and match formats. Again, this is like a tour through different kinds of graphing formats for exploratory data analysis. In this case, I've got this. See this? This is one graph. It's actually on what's called the Y2 axis. That's what this is. There's these points, and they're connected by this line. And this is the mean, all of those. On this axis, the Y1 axis, is a bar chart. I put these two on top of each other. You can graph things on top of each other like this. Go, okay, and what we're trying to do in this particular bar chart is that this is drought, this is like weather conditions, and this is fire. Like, oh, this is a forest ecology. I'm a forest ecologist. Like, we see more fires when there's more drought. A lot of this is kind of messy, but this, except for this one, this looks really clear because this line goes way down here. See this? and this bar goes way up. So this year, which happened to be Dust Bowl era drought, there's a lot of fires. The main point of this figure is just to say you can mix and match formats. Be creative. This is also a mix and match format where you've got Y1, which is this. This is tree ring width. And this Y2 is this bar chart, actually. They're on top of each other. So you can actually use these two to understand visually what's happening with this data set. We might do in the class what's called ordination. I won't go through the entire description of what's happening here. This is a advanced topic in data analysis. We might get to it this semester. A lot of people use ordination for microbial data or like genetic data or like species composition data. Yeah. This is a cluster analysis. I'm really excited about this approach, so it might be fun to do it if I can come up with it in class. So this is like a phylogenetic tree. So if you know what a phylogenetic tree is, like a tree of relationships, but you can use cluster analysis. So this is like the things that are close down here are like these two are really closely related, and this one are much more distantly related. And the strength of those relationships is represented in the structure of this thing. Here's a trick that people aren't aware of. You can use cluster analysis for pretty much anything you do ordination. It's just style, a fad to use ordination instead of cluster analysis. This is called cluster analysis. And in some cases, this kind of visual display is more appealing and more intuitive than this. I encourage you, if you're working with the data set, again, this is an advanced topic, but later in your life, if you're like, I'm doing an ordination because my PI said we should do it with this molecular data, I want in the back of your head to be like, you know what, I could do a cluster analysis. And it wouldn't be that much harder. And actually, it might display the data in a better way. It doesn't have to be phylogenetic relationships. This particular one I created, and these are plots, and the way this thing is structured is based on what plants are in the plots. So it's about the ecological relationship, not the genetic relationship. That's a pretty cool way to do it. I haven't published something like this, but this is this interesting approach. This is called a bubble chart. We might do this in class, but you can imagine some kind of regression kind of approach or a scatter diagram. You size the points based on some other variable. This is a nice way to display data. I'm very pro bubble chart, just to say. This particular one is aspect is which way the slope is facing, and elevation is how high you are on the slope in the forest. And then the size of the bubble is how many herbaceous plants are there, like wildflowers. And so in April, there's a lot overall. So you can quickly see April, there's a lot fewer. It's a lot less than in June. June is the maximum wildflower in the forest compared to August. And you can also see this way from northeast facing slopes, which are wetter if you've ever gone hiking before. Southwest facing slopes are drier, and that there's much less herbaceous cover southwest facing slopes versus northeast facing slopes. So you get a lot from this visually. But your analysis, what the size of that might be microbial colonization, or might be some, whatever. It could be some genetic data, like, oh, these alleles or something. You could chart allele frequency this way. This is just another bubble chart. In this case, I've actually, size matters. Also, the color matters. This is sample diameter, and this is slope aspect. This is a tree ring kind of analysis. I'm asking you here, just don't be boxed in. Don't limit yourself to figures you've seen before. This is totally creative. There's no one has ever made a figure like this before or since. I just invented this because I thought it was a good way to display this data set. And once you get going in R, you'll see it as a sandbox. You can do whatever you want, actually. So I want you to be creative. You can also display data in a geospatial context like this. This is actually a map. And you can do this in R. We won't do it in class. It's too advanced. But these are points, and around those points are these donuts, and the donuts tell you something about whether a plant was there, Americas, in these different years. So you can have a geospatial display that's overlaid with a display of some kind of biological parameter. You can do this with an image of a fly's eye as well. This is literally a forest, but you could do it with a fly's eye, or maybe like, yeah, you can do it with all kinds of things. This does tell us something, gives us information about what's happening on that site. I won't go in and unfold it for you. I've got one last thing to say in this part for exploratory data analysis, which is I want to encourage you all to be careful about black box kind of analyses. You might build a model in your study. If you're a doctoral student, especially, this is an advanced topic, but this class, there's advanced students in here as well. You basically tell your computer to take your data, shuffle, then analyze. You put all the data into a black box and you go, okay, let's see what happens. But I want you to be really careful if you do that. This is what I'm saying about exploratory data analysis. Even a ANOVA is a black box in a way. But if you start doing like, I'm going to do principal components analysis, or my advisor always does this kind of, genetic analysis or something. You of course do that because that's what your advisor does, or the people in the field always do. We always run our analysis through this big complicated thing. However, you as a data analyst are responsible for the data set. Still, you need to know what's happening in your data set. So I'll show you an example. I worked with a modeler on this study years ago, and this was a model she generated. It's a very complicated model. She generated this extremely smooth, predictive model of how much oak, this is a kind of a tree, how many oak leaves are on the forest floor, to how much amber honeysuckle is a kind of a shrub, how much amber honeysuckle is in the forest. So it's like, as you increase the amount of oak litter, the amount of probability of amber honeysuckle goes down, and it's very smooth, and these are very clean. Model depth of 0.5, model depth of 2.2, model depth of 3.0. So this looks like, wow, we're really on to something here. But because I'm the skeptical scientist, and I was also brought up in this exploratory data analysis mindset, which I'm trying to give to you now. Even though I was working with a statistician, I was like, I don't know, man, that forests are never like that. This looks like, forests are crazy. There's all kinds of shit happening in the forest. Birds and deer, soil pH, tree falls. Just like, it's never this clean. And so I did another analysis myself, and I required us to publish both in our study. I think both are in there, which is this. This is the data. These are the data from which that study, or that statistician generated these very smooth lines. This, which is like how much honeysuckle is there measured in the field. And you can see, I rank ordered this. This is going down. How much litter is there? Oak litter. And you can see there is a relationship. It looks like it. Honeysuckle abundance out here is very, very low in areas where there's a lot more litter. So it does look okay. However, look at this one. This is almost the highest bar in the whole thing. And look down here. Well, there's a lot of honeysuckle there. And look at this bar. This is pretty high in the grand scheme of things. And there's a lot, almost a max honeysuckle there. So this is pretty messy, actually. So, this looks very clean because it's been modeled through some black box process. The biological reality is this. That's what we measured. Data from this were used to generate this model. And so, as a data analyst, and this is in a peer-reviewed journal article, we published both figures. I'm okay with having this figure as long as we also have this figure. So a reader can go, yeah, this does look like something's happening, but it's pretty messy too. So that concludes, this was a long lecture, and I did have a glitch there halfway through, but I got it back on track. And Thanks for listening, and we will see you. I mean, this thing. We'll see you in the coding club.